Hi, can you hear me? My name is Peterman. I'm a graduate student in Aerospace, which is between physics and the Center for Systems Biology at Harvard. I'm going to talk to you today about the functional landscape of regulatory RNAs, looking at some of our experiments in and analysis in bacteria. So, start with the simple dogma of molecular biology from DNA, RNA, and then protein. And when we're looking at the regulation of gene expression, some of the best understood systems are transcription factors, and what we're looking at today, or what I've been spending my PhD looking at, is regulatory RNAs, which are ubiquitous in gene regulation and allow the organism to skip some steps that might be required for transcription factors. So it's not surprising that we see these regulatory RNAs abundant across gene expression, but also specifically where fast response is necessary, such as stress response, virulence in specific, many other examples. But to give you a sense of what we're looking at, RNAs, regulatory RNAs in bacteria are typically 50 to 100 bases in length, and they have this stem-loop structure, which gives them some structural flexibility in their activity, and they're found both as more commonly as repressors, but also as activators in some cases. And just to give you a cartoon picture of this, we see the regulatory RNA on the top, RIB, comes in and pairs with the 5' untranslated region of its target side B here, and by doing so blocks translation and also recruits the degradation of this target for non-catalytic negative regulation of this target. We can also see in a limited, but numerous cases, activation where the native secondary structure of the 5' UTR of the gene of interest, here SHE-A, another target of the same RNA RIB, prevents its expression. Both, you see that the start codon is paired to another part of the 5' UTR, and the crucial ribosome binding site is also buried in that tight structure. And here we have the small RNA coming in, and by binding, creates a change in the conformation of that structure, allowing expression and effective upregulation of this target. So the question that we're interested in looking at is, what are the sequence determinants of this process in the cell that allow this function to occur? And the approach we took was SORT-seq. This was a technique that was pioneered by Justin Kinney when he was a physics graduate student at Princeton a few years ago. But to give you a sense of it, what we do is the goal is to characterize many variants of this gene simultaneously using a high-throughput sequencing approach. So we do mutagenesis to the regulatory RNA gene and zap them into E. coli. And because we have a very diverse set of mutants here, they have ranging activity from completely inactive, so this is negatively regulated in green in this picture below. Those white cells are highly active, and the green ones are very inactive. So looking at this through flow cytometry, we have some variability even in a single genotype. So here I have the histograms for the wild types regulatory RNA and some inactive negative control, and we can take this library. So many variants make up this library. They range from active to inactive, and we can sort of sequence this library to quantitatively characterize and on the order of tens of thousands of variants in one go. And just to say, we've done a lot of work on this and have this very adaptable technique that many of you may be interested in talking to us about. And we have a methods paper in preparation that looks at some of the determinants of getting one of these assays to work. But to jump into the results, just looking at a small subset of the variants, just the ones with a single mutation, we can very quickly see that this technique can create a lot of informative data about these regulatory RNAs. So just to give you a guide of what this plot is saying, we have two targets, and along the x-axis we have base position along the regulatory RNA, and the y-axis is the base that is mutated to. And here, blue colored squares mean that the RNA has lost activity in red, and a few red or pink ones means they've gained a bit. And we see that there's certain areas of these that are highly correlated between the two different targets that I told you have completely independent pathways of regulation. So it's likely that these correspond to general features of the small RNA, not per se the interaction with the target. However, in the center, we see target-specific features that are also agreeing very much with what we might expect from seed pairing and those considerations. So informed by sort of this data, we can go forward to the rest of the data and look at the modules that are involved in this process of gene regulation, or what makes a particular small RNA variant work. And the first two would be the folding and unfolding of those two, the first and third stem loops. And so if we just throw this into an RNA folding algorithm and look at variants with only mutations in those regions, we can see that just this folding can tell us a lot about their expression. Moreover, in the sort of target-specific, and just to say this is very consistent between the two different targets. And if we look at the target-specific component, looking at the pairing of the seed and the seed match region on the mRNA target, folding can also give us a lot of information about that interaction, and we can see that by tuning the seed binding energy, we can tune the fold repression of the target by the small RNA. So introducing mutations that increase the length of the seed can actually improve repression, and introducing, say, just a GU wobble will cut it in half but not destroy repression, though many other variants destroy repression. So just these three modules can actually explain a lot of what we see in our mutagenesis data set, where we do an average of two or three mutations to every small RNA molecule. But we wanted to take a different approach, a more sort of naive modeling approach, where we just instead take individual contributions. So let's take each mutation as completely independent and make an independent additive contribution to the activity of that molecule. So here we just use a two-state binding model as a function of this energy, and these are just additive terms in this sequence activity model. So we can see that we get a lot of the information in the data set, much of the variants covered here. But really what's interesting to us is I mean, it's fantastic that this supports us. So here, the orange dots are fixed to the line when we're comparing the model and the measurements. But green are not fitted. Those are variants with 2 mutations, and purple with 3 or more. But in fact, what we're more interested in here is not the points that are close to the line, but the ones that are far away. So once we have this quantitative model for sequence activity, we can also identify where the model doesn't work. So we call these deviations for pairs, we call them interactions. Another word would be intermolecular epistasis here. And we can see that while most variants are near 0 in sort of interaction strength, which we define as the difference between fold change and expected from the model, there are significant outliers, both positive and negative, which we call synergistic or antagonistic interactions. So when we characterize, the next place is where we find these interactions. first place we looked was where we might expect to find them. So we have all this seed pairing happening within the stem loops, and so if you mutate, if you make a mutation on one side of the stem loop, you might expect that to hurt activity, but another mutation on the other side would play a compensatory role. That would be a telltale example of a synergistic interaction. And when we look for such Watson-Crick pairs of mutations, you see exactly that. So this is all Watson-Crick mutations, 1/9 of all of the pairs of mutations in the data set. And we see exactly these where we expect them to in the first and third stem loop from the structure. When we look outside of this subset, we find a lot of stuff that we had no idea we'd find. And I guess I'll draw your attention to one thing in particular that was interesting to us, which is this mutation at the base of the middle stem loop called SL2. And this mutation has synergistic interactions all across the seed region of the sequence. And so the way that we interpret this is that this mutation will allow the small RNA to remain effective despite mutations within the seed. So in another word, it's relaxing the seed specificity of this regulator. And we went and made mutations on the other side to the target and confirmed that that's really the case. So there's something deep going on between this mutation and when we think this stem structure and the specificity of the regulator, which is really key because for every 1 or 2 or 10 targets, you have hundreds of others that you critically do not want to downregulate. This is something we found we're looking to. I'm interested in finding out more from this rich landscape of this rich data set about the landscape of RNA regulators, and we're also interested in looking at the competition. So as I mentioned, this RNA regulator is a pleiotropic regulator. It has multiple targets. So how do these sequences do their work with more than one target simultaneously? Is there some sort of prioritization, et cetera, going on here? So with that, I'll move on to the acknowledgments. In particular, Errol, my advisor, and Anat, who worked with me hard on this paper, as well as others from the Levine Lab, and some advisors and collaborators in Harvard and Princeton. And I'll just also acknowledge the funding, and I'm also giving a poster tomorrow, so check out my poster, and I'll be glad to take any questions.
Can you give priorities to students first? Students? No? All right. So present. Go backward.
Yeah, very nice work. I have a quick question. So you, so far the small RNAs that you presented are all pretty well known. But is part of your also algorithm, are you looking for new ones? Because like you hear numbers like maybe 50 are confirmed, so they've done experiments on them. But like there's several hundred that have been predicted in the
Yeah. Yeah, in fact, every month it seems there are new classes even found, like a whole new class of ProQ associated small. So these are all HFQ associated small RNAs, but they're finding new classes of small RNAs. Our technique here is not so well suited on the experimental side for finding new targets because we have sort of well-tuned reporter systems so that we can do this at such high throughput. However, I do hope that some of the insights that we get from the modeling side can be used to help inform some of the target search efforts. So I think in particular the structural relevance of the first stem loop and also seed specificity are both really important questions because all of the sort of computational target search efforts are riddled with false positives. So you get 100 false positives for every few that you find real targets. So that's absolutely right.
Thank you.
So, is methods, I guess the experimental method, limited to bacteria, or are you going to imagine.
Not at all.
So you can, are you doing this for mammalian cells?
Yeah, so,
where are you in that? You know?
So there have been, in writing methods paper, I've reviewed all of the, I think about 20 papers have been published so far using sort-seq and bacteria, yeast, mammalian cells as well. And the mammalian cells, getting these assays to work quantitatively, I think is a little bit tricky. Some people have tried and done pretty well. And we're involved in some work looking at mammalian transcription as well using sort-seq. But yeah, in principle, this technique, some of the details are a little bit different, but this technique is applicable broadly. And also, I think going back to the earlier comment, this technique is very well, I mean, we started with a very well characterized, or two very well characterized bacterial small RNAs, but this is very well suited to something you know nothing about. So that's some of our future work as well, is looking at that. I know Rob Phillips' lab has been using that to characterize promoters that are completely uncharacterized.
You showed very interesting data, synergistic effects between the C fusion mutation and the mutation with SL272 at the bottom of that. And so what can you do to actually get deeper into the mechanism? Do you have some plans on how to actually examine?
Not in particular. I think one of the people that we've talked to about this is Julius Lux at Cornell, who's doing RNA structure in a high throughput way and looking at some of these variants technique. I think that would be fruitful. I think also characterizing, just because this is just one example, characterizing other examples of what sequence features are governing C specificity would be worthwhile.
To look at other siRNA pairs to see that something like this also pops up.
Yeah. Thank you.